Today I'll be discussing different point mutations within the DNA coding strand. Within this drawing, a protein that is 5 amino acids long is from this DNA sequence. In order for this amino acid sequence to be made, we first need to transcribe one of these DNA sequences into mRNA. If we flip the bottom strand, we see that this strand has a start codon, ATG, where the T will be replaced with a U in the mRNA transcript. This strand also has a stop codon, TAA, UAA for the mRNA transcript, that is 5 codons down from the start codon, which will make the bottom strand our coding strand. Now that we have our coding strand, we will transcribe this into our mRNA sequence labeled below. This process will just replace the T's with the U's. We can now translate this mRNA transcript into an amino acid sequence starting with our start codon, AUG, which codes for methionine. Moving down to our next codons, we have GUC coding for valine, AGG coding for arginine, UUC coding for phenylalanine, AAG coding for lysine, and UAA, which is our stop codon. If there was a mutation to occur where we change the highlighted guanine to a cytosine in the DNA coding strand, this will be an example of a transversion from a purine to a pyrimidine. This mutation would be considered a missense mutation, as a new AGC codon would code for serine instead of arginine, changing the original amino acid sequence. If there was a mutation from changing the highlighted guanine to an adenine in the DNA coding strand, this would be an example of a transition from a purine to a purine. This mutation is considered a silent mutation, where changing one base won't change our amino acid sequence because AGG and AGA still code for the amino acid arginine. If there was a mutation from changing the highlighted adenine to a thymine in DNA coding strand, this would be an example of a transversion from purine to a pyrimidine. This mutation is considered to be a nonsense mutation, as when this thymine becomes a uracil in the mRNA transcript, this now UAG codon codes for a stop codon instead of the amino acid lysine. The result of this mutation would cause a shorter and likely non-functional protein. If there was a final mutation from changing the highlighted adenine into a cytosine in DNA coding strand, this would be an example of a transversion from a purine to a pyrimidine. This mutation is considered to be a read-through mutation, as the original stop codon, UAA, is now coded to a UAC. This codon now codes for the amino acid tyrosine, where the amino acid chain continues to elongate, forming a longer protein chain. A simple analogy to remember these 4 point mutations examples is the idea of making a simple cake with eggs, flour, milk, vanilla, and sugar, our 5 original amino acids. If we were to change our sugar to a brown sugar substitute, or silent mutation, these ingredients would still make our final cake product, even when we change our sugar additive slightly. If we were to change our eggs into a banana substitute, or Missense mutation, these ingredients would still make a cake, however, the cake altogether would be different than the original cake product. If we were to get rid of our sugar altogether, our nonsense mutation, this will cause a change in our ingredients and cooking list, and thus likely make a cake that is not sweet and not good for consuming. Lastly, if we were to change our ingredients list, where we will add more cake ingredients rather than stopping at sugar, such as chocolate chips, icing, and caramel, our read-through mutation, this will cause the cake to have more than 5 original ingredients and thus make a more extravagant cake than the original cake product.